Next slide, please. So our consortium continues to move forward, and we have 55 sites with data released in the N3C enclave, 42 sites deposited data. I just saw the other day we have over 2 million COVID positive patients and a much larger number of controls, and 211 sites have DOA executed. Next slide, please. So, okay, without further ado, Joy, looking forward to your talk.
Great, and thank you, Joe. And so our research was to find out if we could find repositioning targets to impact COVID. And so there are two parts of the research that we had. One was within N3C and one was outside of N3C. So if you go to the next slide. First, before going further, I'd like to acknowledge the team. It was a multi-institution, multi-team effort. We're really appreciative of N3C for the data access, and also to the COVID-19 HPC Consortium and the multiple supercomputing centers that helped us for the molecular simulation compute time access. If you could go to the next slide, please. So the big picture of what we wanted to do in the two-step process for research, we wanted to simulate 1,513 FDA-approved compounds against 11 specific coronavirus proteins and the substructures thereof to find out if there were specific atomic molecular interactions that we could detect to potentially interrupt these protein structures. And the idea was that if we had a short list of candidates that would like to validate it using a patient data set, and that's where N3C came in. So using the compute time that we were allocated outside of N3C and doing the novel quasi-quantum simulation that we've been working on for the past 2-3 years, we found 18 specific drugs that against the 11 specific coronavirus proteins that we simulated that seem to indicate to us a molecular interaction energy that there could be a potential interruption that could happen with these drugs. So after doing this simulation, we had these 18 drugs, and but we needed to validate it, and it's not possible to run 18 different randomized clinical trials. So observational validation was the next best thing. So using a multi-predictor propensity score-based cohort matching, we used the N3C's December 7, 2020 data release, which is about the time that near the time that we wrapped up the analysis part of this project, to see if any of these 18 top candidates from our molecular simulations resulted in a reduced COVID-19 mortality odds, the primary endpoint that we looked at. If you could go to the next slide, please. So the idea is, of course, we have the drug compounds once in active form has a window of opportunity to find a potential cognate protein, and each protein, there has multiple targets for interruption, and there are the existing drugs that target these proteins differently. So, for example, a compound could have an active site interruption, and there are drugs, example drugs that happen for the particular clinical causes they're trying to solve. There could be an allosteric interruption, so not directly on the active site, but somewhere else to have an energy conformation change that results in a reduced activity or a desired activity. And then, of course, regardless of the protein direct active site or allosteric interruption, proteins many times have to come together as a complex for it to perform appropriately. So we could potentially also prevent a complex from forming. And that was the primary idea of why we were doing molecular simulation in the first place, to see if we could in silico detect any of the existing drugs that could potentially interrupt either active site or allosteric or a complex formation interruption using the 1,513 FDA approved drugs. If you could go to the next slide, please. But there are many coronavirus proteins. What we wanted to do, we took 11 of them, 11 that we thought would be more highly likely for a potential effect, to see if there would be an in silico clinical signal that we could detect. And if you go to the next slide, so on the right-hand table here, these were the 11 specific coronavirus proteins we looked at to simulate against, and and existing literature on why some of these proteins are important. And we took each of these protein complexes and separated it out into its constituent substructures, all in silico, and then assessed it at neutral pH. So that resulted in an energy distribution, a topological energy distribution. And from that, the algorithm that we developed automatically selected or determined what would be potential candidates for those energy on that topological energy distribution. So then each of those 1,513 drugs were actually simulated for each of the potential candidates for interruption of each of these proteins. So there's a large computational effort that went through. And based on the results, we found 18 top scoring drugs that in silico predicted that there could be a potential interruption. Now, the next step from here is to actually going into the N3C world, that we had 18 top candidate drugs, and we had to observationally validate these. If you could go to the next slide, please. And next slide. This is just a picture rendering of how the simulation could potentially visualize the interruption. Now, this shows you the distribution of our top candidates, not all the candidates, our top candidate drugs. And as you can see, I want to note on the x-axis, these are some of the top interruption candidates. On the y-axis, it's a score of how likely the interruption is. And here, the lower the score, the better. So you're looking for a lowest energy confirmation. And I want to point out a few things here where the drug metformin, which Dr. Bramant last week also talked about a lot, was our #1 top candidate in terms of viral protein interruption, followed by hydrochlorothiazide was #2, and then amoxicillin was #4, and then later on triamcinolone is somewhere later on in the x-axis. And I want to note these 4 drugs because as we go, look at the results, it'd be important to reflect into. So this I thought was one of the more cooler parts of the project, because this shows you the interaction prediction from the in silico. And then whenever you're looking at a graph or a chart, I look for a cliff, and where does the chart or the graph just falls off? Is there a major change? And you can see the cliff is right about here. And noteworthy here that metformin and hydrochlorothiazide were on the left side of the cliff, which is at the bottom of the cliff, which is what we're looking for. And if you go to the next slide. So, we looked at, did a multi-predictor analysis using age, gender, race, BMI, comorbid, Charleston Comorbid Index, prior hospitalization, access to medication. And it just shows you a distribution of at least across all patients on some of these predictors and across COVID positive patients for these predictors. If you go to the next slide. So based on the multi-predictor cohort analysis, what we found out was, metformin, triamcinolone, amoxicillin, and hydrochlorothiazide, of the 4 candidates we had, all had a statistically significant reduced odds of mortality. And what's noteworthy here is, at least what was exciting to me, was metformin and hydrochlorothiazide were our top 2 candidates. And consequently, they were also the 2 of the top candidates that resulted into a statistically significant mortality odds reduction. So we looked at this results preliminarily, and then thought, well, maybe metformin's there because these people have diabetes under control, and that's where perhaps is the causative effect, and not the and perhaps not the direct protein interruption. So the next analysis we ran, well, what if we looked at the use of metformin across non-diabetic patients? and to see if we could also detect a signal there. And we also could detect a signal there for a reduced mortality odds on metformin use in non-diabetic patients, or what we think are pre-diabetic patients as well. So which is also I thought was a nice validation of our of the research with it. And it's if you go to the next slide, please. So, the discussion, points that we had, and after, after this research was done, is, this is actually very good. And we were actually quite encouraged because there were other research groups that were also looking at the effect of these drugs from independent of our findings. So, for example, Dr. Bramant mentioned her group's work on metformin, and they're looking at outpatient use of metformin. There's a separate group that's looking, it has an RCT on the hydrochlorothiazide and its effect on COVID-19, which I thought were also perhaps quite validating of the in silico results that we found and validated using N3C. If you go to the next slide. So, well, one of the things that happened after our preprint went out is a research group from India, in Pune, India, contacted us to see a potential, if because of the situation there, a potential for an RCT there on use of metformin in non-ICU admitted COVID-19 patients. So we drafted a protocol and looked for funding for them. And got some funding in place, but not enough to cover an RCT. We're about $170,000 short. So if anyone has any ideas on where funding, if we could help them obtain additional funding to do the RCT, that would be really cool. If you could, no meeting is complete without an ask for money. And so if you have some comments or thoughts, that would be great. You can message me or send it via chat. I also want to make a disclaimer that my research group doesn't have any financial interest in any of the drug candidates we found. So there's no financial interest on this from us. We're really appreciative of the data access and the rich tools that N3C has provided, along with all the support and training. This research wouldn't have been possible without the support from N3C. So if there's any questions, I'll be happy to answer, but that's around the end of the presentation from my side.
Thank you. Questions? No, that's very cool research, and I think a lot of people had informally discussed doing this sort of combination of mechanistic and population research, but really very few people are able to pull it off. So my kudos to you. What are so maybe you could talk a little bit more about what else you'd like to do using these sorts of mechanisms. For instance, have you given thought to potentially targeting potential variants that might arise with particular drug treatments?
So yes and no, as in we've given thought to it. We haven't spent much time or effort on simulating against the new viral proteins from the variants. My research team, one of the other diseases we're looking at is dengue, to see if for which there is also no treatment, to see if there's a reposition drug candidate we could find. But beyond this particular fortunately, the impetus behind further simulation for COVID-19 is against variants, at least, is not as predominant here in the US. I think as the numbers in the US are going down. So a good question, but we are not planning to simulate further proteins for coronaviruses, if that answers your question.
Mm-hmm. And certainly all being well, we've got mechanisms for generations of vaccines against variants. So right. That makes sense. Okay, well, thank you. This was a great talk. Appreciate it. Okay, so Christine, on to you. Look forward to what you have to say.
Okay, thank you. Can I have the next slide, please? Thank you. So today I'm going to present to you the framework, the governance framework that we established for N3C. And I have 4 points I wanted to present. One is, giving you an overview of what was the N3C governance. I want to highlight the community guiding principles. Then I'll describe how the data can be contributed and accessed. And lastly, I'll talk about the attribution and publication guideline. Can I have the next slide? Okay, so, it was April 2020, and we needed to develop a governance framework to enable really the rapid research on COVID-19. The speed was critical, right? We needed to have fast and easy access to the data exclusively for COVID-19 related research. But we also needed to protect patients' privacy and ensure the data security. So the data governance was developed in partnership with NCAT that act as the steward of the data and and a trusted third party. Can I have the next slide? The governance is really this partnership between the N3C community and NCATs, and each have their really a different role that is kind of complementary. N3C community will develop the community norms. They enable the team science and created some work stream with different expertise and really promoted the joint publication. NCATs focus on controlling and overseeing the data enclave, oversee the contribution of the limited data set and the data usage. So this partnership between N3C and NCATs in data governance is illustrated by the work that the governance work stream has done in the last 14 months, focusing on establishing the principal policies and best practice for N3C, create the function, the role, while clarifying who can do what and how the policies and strategy can be implemented, and then design the infrastructure and procedure for the control and audit of the activity on N3C. Can I have the next slide? So it's now, and here is kind of a summary of what the work stream has accomplished. All of the policy, or most of the policy, are posted on Zenodo, so you can look at them. They are available. So the community and NCAT, partner and author different part of the governance. So there is some policy that have been authored by the N3C community, other that have been authored by NCATs, and some that have been authored by this combined partnership. For instance, the N3C community developed the community guiding principle that are the, clarifying the cultural and behavioral norm for participating in N3C. There is an attribution and publication policy that outlined the requirement for open attribution that is supported by the publication committee chaired by Mary Salt. We created for a single IRB for facilitating the data contribution. And early on, John Hopkins agreed to act as the IRB of record for data contribution. This was really very helpful to use this single IRB. However, it was not mandatory. Sites who wanted to contribute their data under the authority of their own IRB could do that. And then we established the community response team to address different issues. NCATs and in partnership with the community, right? The work stream also developed a user code of conduct to articulate, the user agreement to take responsibility and about accessing and using the data. There is a data transfer agreement that was developed to that needs to be executed between the sites that are going to contribute the data and NCATs, and that's at the institutional level. A data use agreement that needs to be executed between the institution that want to have access to the data and NCAT. And there are different mechanisms like the data use request for individual to request access to the data. And those requests are evaluated by an access committee that is controlled by NCAT. And there are new policies about bringing public data into the enclave, policy and committee reviewing the download. And currently we are really focusing on hashing and data linkage policy. So that's kind of a lot of the work that we have done in the last 14 months. Can I have the next slide? So I wanted to highlight the community guiding principle. The first version of those principles was posted in June 2020, and we just revised the guiding principle, posted an update where we added the last principle of mutual respect and highlighted some communication etiquette. So those 7 principles are, emphasizing partnership, inclusivity, transparency, reciprocity, accountability, security, and mutual respect. So really, an N3C is a community member, trusted partner. Anyone is welcome as long as they agree with all of the terms. The project must be posted so that enables and elicit more collaboration. There's the expectation that analysis results are going to be shared with the community, that every member are taking responsibility for their action. The work is done in a very secure environment, and activity are being recorded. They can be audited. And the expectation of behaving professionally. Can I have the next slide? So in addition to those 7 principles that are listed in the community guiding principle, there are different statements that highlight expectation. The ethics statement really emphasize that the work that is being done with the data needs to really consider patient and the public first. It's important that user of the data consider the effect of their research on the vulnerable population. There is the expectation that, of course, there is contribution must be acknowledged, must be recognized, the expectation to uphold scientific integrity and bolster public's trust. There is a diversity and inclusion statement that highlight the fact that N3C is a community that honors diversity and broad diversity. There's a communication etiquette that we just added to really reemphasize that we want to promote positive interaction, and that if there are any issue that occur with any large community, that the community is going to use community arbitration and work toward positive outcome to resolve any issue. There is the community response team that has been created that can receive any issue or any and address any complaint. So I know I'm going fast, and I wanted to move on from the guiding principle that really control the culture and behavioral norm to what does it take to contribute data and access data. And because the slide is a little busy, I split it in 2. And let's talk first about the data contribution. So institutions who want to contribute data need to obtain approval from the IRB or the single IRB of record from Johns Hopkins for the data they are going to contribute. The data is a limited data set, so sensitive data that has some identifier. So therefore, it's essential to have IRB approval for that contribution. But institutions also need to sign or execute a data transfer agreement with NCAT. NCAT also obtain, or NIH obtain IRB approval for the N3C platform. The data that is contributed is getting harmonized and then is split in 3 different tier of data, as you know, synthetic data, de-identified data, and the limited data set. Can I have the next slide? And the next one. Thank you. So access to the data is more require the user to be from an institution that has signed or executed the data use agreement, and the user requester of the data needs to register on N3C and agree with the community guiding principle. They must make a request and send their request to NCAT. The request includes agreement to the code of conduct, declaring the intended data use and who are the different collaborators that will need to have access to the data, and attesting to having completed the security training and age security training and the human subject protection training. The data use requests are then reviewed by the Data Access Committee, currently under the control of NCAT. It's an NCAT Data Access Committee. And if the user wants to have access to the limited data set, they also need to have proof of IRB approval for their internet data use. The Data Access Committee reviews all those requests and then determine whether the level of data that is requested is appropriate for the internet data use, and then release the data to the user. Can I have the next slide? Okay, so you agree to all the term of the community, you agree with the principle, the code of conduct, you've obtained access to the data, you are doing your research, and now the expectation is that you are going to publish and share your result and publish the data and acknowledge all of the contributor to the research that you have completed. There's the attribution and publication committee. There is a guideline for attribution and publication, and the support of the publication committee to review the different publication, different manuscript or any communication out of N3C. The publication committee has a role to facilitate the attribution of the different contributor, troubleshoot any authorship question, track the output of the N3C and promote and disseminate the information, promote awareness of the work that has been done with the N3C data. The publication committee has worked really hard and revised over the last 14 months how to best to acknowledge the contribution of the different experts that participate in the work that is being done. And there is 3 levels of really recognition that has been the committee that agrees on and is using. First is the masthead authorship, that is, people having their name in the byline of a manuscript. And then there is the block authorship that is listed as N3C consortium in the byline. And this N3C consortium includes people who have contributed significantly to the work, but are not necessarily having participated to the elaboration of the hypothesis and may not or wrote necessarily the manuscript, but whose contribution was essential to accomplish the work. And there is, of course, acknowledgement, that is, standard acknowledgement statement. The masthead author and the block author need to comply with the ICMG standard, and their contribution and their name is going to be indexed in Medline. Can I have the next slide? So I wanted really quickly to clarify what the ICMG requirements are for authorship. ICMG has 3 different category of activity that they have identified for manuscript. There is the first step, which is the conception and design, and they are requiring that author either participate in the data acquisition or the analysis and interpretation of the data. there is a second step that is drafting the manuscript or revising the manuscript. And so author need to have contributed to the manuscript. And the third step is having final approval of the manuscript. So according to ICMG, an author needs to have contributed to those three steps. If someone contributes less to than those three steps, then they are a contributor and not an author. So currently, N3C is and the publication committee is really supporting and reviewing that author are contributing to manuscript according to ICMG, but we have a lot of discussion about whether those standards are appropriate for conducting team science the way N3C is doing. Can I have the next slide? Thank you. So if you want to learn more about the data governance, the cohort paper really describe the different tier of the data, how the data is has been organized and describe really the governance framework in a little bit more detail. You can take a look at that, but you can also feel free to join the partnership and governance work stream. You can find more information on the Slack channel, the group. Know that the work stream is meeting every Friday morning at 7 a.m. Pacific, 10 a.m. Eastern. And if you're interested, please join that work stream. And if you have questions about the publication committee, you can email Mary Salves, who's chair the committee. And I think that's my next slide. I think the next slide is just a thank you slide. So if you have any question, I know it was fast, but hopefully that gives you a sense of what the governance work stream has been working on.
Great. Well, thank you, Christine. This was very interesting and very important. Does anybody have any questions? You put them in the chat if you're attendee.
I believe there's one question. It's from Hiroki Morizono. Currently, what fraction of CTSA hubs are contributing their data into N3C?
How many are contributing data? I think that's, I don't know how many are.
We had the slide up front. Maybe go back to the slide up front. Let me see if I can pull up that slide.
Well, I guess we have a number of contributing sites. I don't think it's broken down by CTSA.
Yeah.
The number of contributing sites. I think we have 88 sites that have signed the data transfer agreement. How many have actively provided data? I think it's 40, or low 50s maybe, but
55.
Right, exactly. So those are the statistics that we have.
Yeah.
So there's 88 sites. So we don't. It's not completely clear what fraction of them are of CTSA sites. That is, I guess they're 60 odd CTSA sites. So they're more larger number of sites have executed DTAs than there are CTSA sites. I imagine a pretty substantial majority of CTSA sites are now contributing data. That's the impression I got. COVID dashboard. Okay, so I think.
So if you figure or filter the contributor table by CTSA and available, you get 38 sites. So roughly 2/3 of the consortium has their data online and available.
So Christine, so you and Mary have certainly done a fantastic work in sorting out the whole very important issue of how people get acknowledged for their contributions. Can you reflect on what some of the challenges have been and what you think might be done in the future along these lines? It's obviously a critical issue given the number of people who contribute in so many different ways.
Well, well, first of all, we started, we wanted to recognize everybody who participated in any manuscript, right? So we wanted really to have authorship that was really encompassing everyone. To be able to do that, of course, we had to distinguish the author that have work on the main hypothesis of the manuscript and written the manuscript, and then create this other authorship tier that is this block authorship. What we found was that block authors still need to read the manuscript, they still need to agree with the manuscript, they still need to indicate what was their contribution. So we really didn't want to have open authorship for everybody. You still need to be involved in the work to really deserve to be an author. Establishing those criteria for what people have contributed to clarify the whole process of indicating, confirming what people have done, having the author being comfortable with the idea of block authorship. Right now what we are seeing is that there is no easy mechanism from from the publisher to be able to easily upload those block author and the dozen and dozen of block authors. So there is a little bit of frustration in terms of the logistic of creating those block author. It's not really in terms of the issue of whether or not to recognize people, but it's really implementing those block author has been a little bit challenging, and getting the indexing in Medline.
Yeah, I'd imagine the indexing would be certainly a major challenge, because
Yes.
the whole issue of publishers interacting with Medline for all of us who need to show that our publications are appropriately recognized by so that's very tricky.
Yeah, it's been taking months and months of email back and forth for some of the publication, and it's still not resolved. It's difficult.
Yeah. No, that's great.
But it's our goal, right? Our goal is to reach that level of recognition.
No, and this seems like, this is really a sort of wonderful prototype for team science, certainly in sort of in these sorts of areas of medicine.
Yeah.
So thank you. No, this is great. So we had a question in the chat for Joy around I think disambiguating or deconvoluting impacts of metformin with other medications. So patients not treated with metformin are treated with other drugs. Patients treated with metformin also had many other drugs. How to adjust for patient selection in this model?
So good question. So our primary approach for this was to rely on the comorbidities and the Charlson comorbidities and the specific comorbidities to distribute out the use of the drugs in terms of our matching. We did not look at specific drugs that are taken in combination with metformin to see if those were matched. And for as that was not the approach that we followed, but it is, if we had more time, and I think that would have made legit, perhaps the study a bit stronger. But that's a good question.
Great. Okay. Fantastic. So any other questions or thoughts?
Joel, we have a question from Mary Saltz in the Q&A.
Okay. Q&A. Okay. So Mary Salt says, what are the main barriers to getting 100% participation from CTSA sites? So anybody, Christine? Any thoughts?
I think some sites are still concerned about providing their data and losing control of their data. But that's, I mean, that's my assumption. I haven't really, I haven't evaluated. I have not really reached out to the CTSA side that have not contributed data to try to ask them what are really making them hesitate. I don't know if somebody else has a sense of what is making people hesitate to participate.
I don't know. I think Ken was on the call. Maybe Ken might have an opinion on it.
Ken was highlighting that there are more than the CTSA hub that are participating, right? We have federally qualified health centers that are participating to, that have contributing data, and OCHIN, for instance, also has contributed data. So it's not the CTSA exclusive network. There are other non-CTSA participants. But why would not all the CTSA hub participate? I think it's probably either lack of resource or the concern about losing control over the data.
But certainly it's impressive that there's so many sites that aren't CTSA sites contributed. I guess it's at this point about half are CTSA sites, which is actually really, I mean, because most sites aren't CTSA sites. So that's really fantastic. That's so I see a comment from Dave Eichmann. There are a number of extensions of current mapping schemes planned that will drill down to a finer granularity.
That's in response to Ken Wilkins' comment just above it.
Okay.
We've done some prototyping of census data mapping and so forth. We haven't fielded anything publicly yet.
Mm-hmm. Yeah, no, I mean, there's certainly a huge potential, and obviously there are a bunch of N3C people, social determinants of health, have looked at mapping, and that's something actually we probably can do a lot more of. So, so wonderful. Okay, another message. Okay, Joel, could you invite someone from the harmonization domain team to talk about submission problems with some N3C sites, such as some sites do not submit death data and visit start and end date data. This is from Fadil Alqua. Yes, I think that's a great idea. I think that's a great idea. So we'll discuss how to do that. I think maybe using this meeting to close the loop on harmonization, as well as actually unit harmonization and quality control, because we actually in that stage of things, when we look to see look at different sorts of data such as vitals and lab data, and look at essentially correcting or normalizing units. There's, I think, a feedback mechanism to the sites that exists, but that could probably be strengthened. We managed to figure a fair amount of this out, but there's probably a lot to be said for sort of iteratively doing that. So I think that's a great suggestion in general, to use this meeting, at least as a high level mechanism for this. Okay.
There's a question from Peter Robinson a few messages back.
Okay. Peter Robinson. Okay, it seems that the effect of metformin is as a plain and anti-hypoglycemic, or by another mechanism. Can we sort that out? Do you have any thoughts, Joy, as to?
Well, I think we tried to alleviate that a bit by running the analysis of metformin and also hydrochlorothiazide on non-diabetic but patients that are taking metformin, and which is prescribed for non-diabetic patients, which presumably would have their blood sugar in control because they're pre-diabetic. But short of doing perhaps a more involved in vitro in vivo models, I think it would be harder to tease that out from in silico.
Okay, no, that's I'm working outside and somebody offered me a pizza, but it's the wrong house. Okay, that's always exciting. Apologize. Okay, that's an interesting thing, of course, about drug, medication effects, to what degree, which I think is in most cases of the sort substantial that you're dealing with a effect of the medication that was not actually the intended mechanism. So specifically, then, did you happen to look at controlling for hemoglobin A1C in impact of metformin?
So we looked at controlling for hemoglobin A1C, but based on the data release that we used, the level of population of that data was not sufficient for us to use that as a predictor. So we used the clinical diagnosis of type 2 diabetes as a predictor, as opposed to a A1C level based approach.
Yeah. No, that's very interesting. Okay, let's see. Am I missing anything else? So we have Ken Wilkins to panelists and attendees. Excellent, many thanks. We'll look further into tracking these exciting developments of interest to BCOR Trust Fund project, looking to partner with CMS on its data. Thank you. So great. Well, these are really excellent presentations. Appreciate everything. So if there's no other questions, we'll give 10 minutes back. Okay. Well, thanks, Christine and Joy and everybody for joining.
Thank you.
And good conversation. Thanks. Bye-bye.
Thank you. Bye.